Welcome to this Bioinfobites explainer, Bioinformatics, Decoded. Today, we're jumping right into maximum parsimony versus maximum likelihood. We're going to break down the two most powerful methods for phylogenetic inference, because honestly, understanding how to build these evolutionary trees is the absolute bedrock of modern biology. Let's get right into it.
So here's the wild thing. For any given set of sequences, there are thousands, and I mean sometimes literally millions, of possible phylogenetic trees. To put that into perspective, if you're looking at just 50 species, there are actually more possible evolutionary trees than there are atoms in the known universe. Crazy, right? So how on earth do you find the best one? You can't just guess. You need some highly specialized, rigorous methods to find that optimal arrangement. Today, we're breaking down the two absolute heavyweights of tree building that solve this exact problem.
Let's start with maximum parsimony. Think of this method as applying Occam's razor to evolution. It basically says the best tree is the one that requires the absolute minimum number of evolutionary changes to explain the data you're seeing. It's looking for the simplest explanation possible. It works by finding the tree topology that minimizes the total number of mutations, or substitutions, from a common ancestor to generate your observed sequences.
Now, here's a key detail. Parsimony doesn't just treat all sequence data equally. In fact, it actively ignores unhelpful data. When you look at a sequence alignment, you'll see what I mean. Invariant sites, those spots where all the sequences are exactly identical, ignored. Sites where only a single sequence differs, which are called automorphies, those are completely ignored too. Parsimony really only cares about what we call informative sites. These are positions where at least two different character states are each shared by at least two sequences. That's where the real signal is.
But as you might guess, this kind of simplicity definitely has its flaws. First off, parsimony frequently hands you multiple, equally parsimonious trees, which just leaves you with ambiguous results. And second, there's this massive pitfall known as long branch attraction. Because parsimony generally assumes equal rates of sequence change across all branches, fast-evolving lineages get incorrectly grouped together. It's an artifact. They get grouped just because they've accumulated so many mutations, not because they're actually closely related. If you're studying fast-mutating viruses, for sure, this artifact can completely derail your research. So what's the alternative? Enter maximum likelihood. This method takes a completely different, mathematically rigorous statistical approach. Instead of just counting steps, it asks a probabilistic question. Given this specific tree topology and an explicit model of molecular evolution, what is the exact probability of observing the sequence data we actually have? The tree that maximizes this probability is the winner. It explicitly models how DNA or proteins actually evolve in the real world, accounting for things like multiple substitutions at the exact same site and rate variations. Now, because maximum likelihood explicitly models real-world evolution, it is computationally intense. We're talking about requiring powerful, dedicated software like RAxML, IQ-tree, or PhyML. But how do we actually know the resulting tree is reliable once the computer spits it out? Well, we use something called Bootstrap resampling. Basically, the software rebuilds the tree hundreds of times from randomly resampled data columns. If a consensus branch appears more than 70% of the time during this rigorous testing, it's considered highly supported and reliable. So let's put them head to head. Parsimony is fast, it's intuitive, and it seeks the fewest evolutionary changes. It's totally fine if your sequences are really, really similar, but it is super vulnerable to artifacts like long branch attraction. On the flip side, maximum likelihood is computationally intense and noticeably slower. It also relies on having accurate mutation models, but its robust statistical rigor makes it the absolute gold standard right now for most modern bioinformatics analyses. To wrap this all up, parsimony equals the simplest explanation. Likelihood equals the most probable explanation. But I'll leave you with this to chew on. As our datasets rapidly scale up to include millions of entire genomes, will even our absolute best likelihood models be able to keep up computationally? Or are we on the verge of needing an entirely new mathematical framework for evolution? Think about it. Next time we're diving into the fundamentals of genomics. See you on Bioinfobites.